Explain how the structure of starch relates to its function.
Starch is a polymer of glucose and glucose is the energy source. Starch has a branched structure so that they can be easily hydrolyzed to release glucose. Starch has a compact structure so that more glucose can be stored. Starch is insoluble, so it will not affect the osmotic potential of the cell.
Explain the arrangement of cellulose microfibrils and how they contribute to the physical properties of cell wall.
Cellulose molecules can be organized into microfibrils. Layers of microfibrils are arranged in a mesh. Hydrogen bonds in cellulose microfibrils and secondary thickening add strength to the cellulose molecules. Cellulose microfibrils are held together by pectin.
How can cells in the morula have the same genes but develop different structures and functions?
Differential gene expression occurs by epigenetic modification or DNA methylation or DNA acetylation. Only the needed genes are activated, switched on. Only active genes are transcribed into mRNA and translated into proteins. The proteins modify the cells.
Differentiate 1. Totipotent cells, 2. Pluripotent cells and 3. Somatic cells.
Totipotent stem cells, an undifferentiated cell which can form any of the different cell types needed for an entire new organism. Pluripotent stem cells, an undifferentiated cell that can form most of the cell types needed for an entire new organism. Somatic stem cells, undifferentiated cells found among the normal differentiated cells in a tissue or organ that can differentiate when needed to produce any one of the major cell types found in that particular tissue or organ.
Explain the factors affecting the properties of an active site of an enzyme.
The primary structure or R groups of protein. For example, there are polar, non-polar, hydrophobic, hydrophilic R groups. 2. pH. It affects the ionization of R groups. 3. Temperature. It changes the shape of the active site. How the cells of morula change as they develop into blastocyst cells.
Morula cells are totipotent whereas blastocyst cells are pluripotent. Differential gene expression occurs by epigenetic modification or DNA methylation or DNA acetylation. Only active genes are transcribed into mRNA and translated into proteins. The proteins determine OR change cell structure.
Describe the functions of pits in xylem vessels.
Movement of water molecules and mineral ions between adjacent cells. OR lateral movement of water.
Define the term niche. Role of an organism in its habitat.
Explain the limitations of diffusion in providing cells with glucose.
Diffusion is too low to supply the cells with glucose. Glucose cannot enter without a carrier protein. Diffusion distance is too great.
Name the two stages in protein synthesis in the order they occur.
1. Transcription. 2. Translation.
Explain how a lack of magnesium ions could result in yellow leaves and reduced growth.
Magnesium ions are needed for the formation of chlorophyll. Yellow leaves are due to their fewer chlorophyll. Reduced growth is due to reduced photosynthesis, reduced glucose.
Describe the processes that would have occurred before and after stage 2 in a drug trial. Stage 1 is testing on healthy volunteers WIYHOUT the disease. Stage 3 is testing on a large number of patients, 1,000 to 3,000. Double-blind trial, analysis of results with appropriate statistical test. Identification OFAPPROPRIATE concentrations, dosage.